artificial breeding. Artificial breeding, also known as controlled breeding techniques. These techniques are in contrast to natural breeding, inbreeding and outbreeding. Here, under controlled conditions, the animals are allowed to exhibit reproduction. There are two common methods. Two common methods that are followed. One is artificial insemination. Artificial insemination. Here, sperm sample collected from a strong, healthy male. Sperm sample that is collected from a strong, healthy male is introduced artificially into the female reproductive system. There are two possibilities here. The sperm sample collected from the strong and healthy male may be immediately used. It is just collected and immediately used for insemination. This is one possibility. Or the sperm sample may be frozen and stored in what are known as sperm banks and used as and when necessary. Another method that is followed in artificial breeding is called multiple ovulation and embryo transfer. Multiple ovulation and embryo transfer, commonly abbreviated as MOET. Here, the healthy female is given injection of FSH or FSH like hormones. The idea is to induce maturation of many follicles. Normally only one follicle matures for each estrus cycle, but here by giving an injection of FSH like hormones, many follicles may be made to undergo maturation. So 6 to 8 ova, 6 to 8 ova are released for each estrus cycle. Each estrus cycle. Again, in a normal situation, a cow would produce only one ovum per estrus cycle, but 6 to 8 ova are made to be released for each estrus cycle. That is why it is called multiple ovulation. These ova are subjected to in vitro fertilization from a suitable sperm sample under laboratory conditions, and the zygote is allowed to develop. The zygote is allowed to develop under controlled conditions. When it reaches the eight to 32 cell stage, 8 or 16 or 32 cell stage, the embryo is introduced into the uterus of a another healthy cow, which is now functioning as the surrogate mother. Remember, all these techniques we have studied with reference to human beings also in reproductive health. They were initially developed for cow. The genetic mother, so surrogate cow gives birth to the young ones. The genetic mother is again used for multiple ovulation. So one single healthy female can be used for developing, for getting many ova, each of which can be fertilized, and the embryo can be introduced into the uterus of a surrogate cow. So this method is called multiple ovulation and embryo transfer.